Okay, in this video, we are going to start talking about the actual process of DNA replication in both prokaryotes and eukaryotes. And we're going to see that the process is actually very similar between the two, but there are a couple of differences that we will want to take note of. So we got a lot to get through, so let's go ahead and get started here. Now, the first thing that we want to kind of do here is, at this point in our study of the history of experiments that told us a lot about DNA structure and function, we of course know that DNA comes from the nuclei of eukaryotic cells, and we know these are the phosphate-rich compounds that Meisner was able to isolate. We know that DNA is, of course, the hereditary molecule, as shown by several experiments, include those very important ones that from Hershey and Chase. And then, of course, the work of Watson and Crick told us that DNA structurally is a double helix that involves base pairing between A and T, C and G, right? Okay, so at this point, the one thing that we really don't know that's really important pertains to DNA replication. We don't know exactly the manner or mechanism by which DNA replication occurs. So, of course, like we said, Watson and Crick's work of discovering that DNA is a double helix, that was important for figuring out kind of that DNA replication, which we found in previous chapters, which happens during S phase. This is something that we need to kind of be focused on, right? So, because of the nature of complementary base pairing, basically it was speculated that each of the two strands could then serve as a template upon which a new complementary strand could be made. And that makes a lot of sense, because if you think about the idea that adenine always binds with thymine and cytosine always bonds with guanine, then each strand should have a sufficient amount of information to direct the synthesis of a new complementary strand. There's not really any mystery involved there. But like I said, the mystery at this point is actually how does that actually occur? So scientists were able to come up with three possible models for how replication could occur. First, you have a conservative model in which both strands do serve as templates for new DNA replication, but the two original strands stay together, and then the two newly synthesized strands are base paired together as well. The semi-conservative model, in which the two original strands separate from each other and then never come back together, so the new strands would then pair up with each of the original strands. And then finally, you have the dispersive model, which, knowing what we know now, doesn't seem very plausible, but this would involve newly synthesized DNA being intermixed with the old strands. Now, distinguishing between these three possibilities ended up being very, very difficult, which is why the next experiment that we're going to discuss, which was performed by Meselson and Stahl, is sometimes called the most elegant or most beautiful experiment in biology, not because of like physical attraction or any other reason why you would normally use the word beautiful, but for how elegant its design is and how perfect it is in distinguishing between all of these possibilities. So like I said, to help us determine which was the correct method, Meselson and Stahl designed this most beautiful experiment in which they grew E. coli, a type of bacteria, in a growth medium that has the heavy isotope for nitrogen, which is nitrogen-15. And since we know nucleotides and nucleic acids contain nitrogen, the idea here is that if you grow the bacteria for many, many, many generations in this growth medium, for which the only source of nitrogen is heavy nitrogen-15, then after many generations, 100% of the DNA in these bacteria should be heavy in nature. So then, after about 14 generations of replication, so if each generation lasts about 20 minutes, then that would be a little less than 5 hours. So after 5 hours, if we then take the bacteria out of the heavy nitrogen medium and switch them over to a growth medium that has nitrogen-14, which is the lighter and much more abundant isotope of nitrogen, then from here on out, any DNA that we synthesize in the future is going to be lighter than the DNA that already previously existed. So the idea now that as soon as we swap over to this new growth medium, the DNA that these bacteria currently have on hand is going to be heavy. It'll have nothing but nitrogen-15 in it. So newly synthesized DNA is now going to have N14. So we should be able to distinguish between these three different methods of replication based on kind of the dynamics of how nitrogen 15 and nitrogen 14 work together between the new and the old strands. So what they actually saw is that after the first light generation, so after the 15th generation, all of the DNA, so 100% of the DNA became lighter than it was before. So this, in essence, eliminated the conservative model from consideration, because if the conservative model was true, then there still should have been quite a lot of heavy DNA and maybe just a little bit of light DNA on top of that. But the fact that all of the DNA became lighter indicated that the correct model was either semi-conservative or dispersive. And then what they found is that after several more generations, this light DNA that we see after one generation here, after the 15th generation, we're going to call that the intermediate DNA. It became more and more scarce, and then more and more of the DNA was even lighter than that intermediate DNA was before it. So we'll see a figure on the next slide that illustrates this pretty nicely. But basically, all of this pointed towards the semi-conservative model being the correct one, and not the dispersive or conservative models. So this illustrated figure here kind of gives you the idea of what is actually happening. Like we said, for 14 generations of E. coli growth, we grow the bacteria in growth medium containing nitrogen 15. So this means that if we isolate the DNA and then isolate it by putting it through a cesium chloride centrifugation gradient, then all of the DNA should sediment as a heavy molecule, as you can see here. So the idea with this centrifugation experiment is that especially heavy molecules should sediment further down in the tube, and light molecules should sediment further up in the tube. So after 15 generations, we can see exactly where the heavy DNA is sedimenting. But you can see that after the 15th generation, so after that first generation of switching over to nitrogen 14, you can see that 100% of the DNA becomes lighter than the DNA that came before it. And again, this is all we needed to eliminate the possibility of the conservative model. So then, after 2, 3, 4 generations, so after multiple generations of being in 14, you can see that this intermediate DNA starts to separate into the intermediate and a much lighter DNA. So based on the way the numbers work out, after 2 generations, it's a 50-50 split. After 3 generations, it's a 75-25 split. After 4 generations, it's an 88-12 split. You can see that after multiple generations, the DNA starts to become lighter and lighter and lighter, and the number of strands that still have nitrogen 15 in them become fewer and further between. And that would not be the case, or at least the numbers wouldn't work out exactly like that, if the dispersive or conservative models were correct. So basically our conclusion from this beautiful experiment is that both strands of DNA become unwound from each other during the replication process, and then they separately serve as templates for the synthesis of new complementary strands, and the two original strands will not come together again. So what the semi-conservative model ultimately tells us is that by unwinding the two original strands, the newly synthesized strands will become complementarily base paired to the old original strands. Okay, so now that we have that bit of historical context out of the way, let's talk about a couple of concepts associated with DNA replication. So first thing we need to do is we need to kind of re-examine a couple of things pertaining to nucleic acid structure. So you'll recall from chapter 3 that a nucleic acid is formed by joining the 3' hydroxyl of the pentose sugar of one nucleotide to the phosphate attached to the 5' carbon of the pentose sugar of a different nucleotide. So what the enzyme is going to do in catalyzing the dehydration synthesis reaction here is form a covalent bond between the phosphate and the hydroxyl of two different nucleotides. So the 5' phosphate and the 3' hydroxyl. So what this is going to mean is that no matter how long your DNA or RNA molecule is, every nucleic acid is going to have two different ends to it, a 5' phosphate end and a 3' hydroxyl end. So this is going to bring up a couple of important points. Number one, anytime you are expressing the sequence of nucleotides in a nucleic acid, you always do so from 5' to 3'. Same way you're writing a word in the English alphabet, you always write it in a particular order. You never write it in the reverse order. So the sense of what a nucleotide sequence tells you will only make sense if you're doing it from 5' to 3'. And then what we're going to see soon, something that actually is going to greatly complicate the process of DNA replication, is that when DNA is replicated, it is always done unidirectionally. It's always done from the 5' end towards the 3' end. So a DNA strand will grow from the 5' end towards the 3' end. So another complicating factor is that if you look here at the two complementary strands of DNA, they are anti-parallel to one another. What that means is that they are moving in opposite directions. This strand here, you can see its 5' phosphate is here, and then its 3' hydroxyl is down here. So 5' to 3', but look at the opposing complementary strand. It runs the opposite direction. Here's your 5' phosphate here. Here's your 3' hydroxyl up here. So this is why we use the term anti-parallel to talk about two complementary strands of DNA. They are parallel, but they're pointed in opposite directions. And like I said, this is actually going to complicate the issue of DNA replication, because when we unwind these strands of DNA from each other, when we start synthesizing our new complementary strands for the two template strands, the DNA replication is going to have to go in opposite directions too, and this is going to cause some a little bit of awkwardness in how we actually get this to occur. So you can see for this strand, DNA replication is going from 5' to 3' this way towards what we call the replication fork, and then for the complementary strand, the replication is going away from the replication fork. So that, like I said, that's going to cause some complications that we're going to have to sort out eventually. Like I said, since DNA replication always happens from the 5' end to the 3' end, and since both strands are going to serve as templates for new strands, this anti-parallel nature, this is going to cause us some problems that that can't necessarily not be solved, but like I said, we're going to have to kind of rack our brains about how we're going to fix that problem. Okay, so what we said before is that there are 4 things necessary for DNA replication to happen in a laboratory setting, meaning outside of the cell and in a test tube. If you want to replicate DNA in a test tube, you have to have template DNA, so you have to have some source of DNA that you want to make a copy of. You have to have raw deoxynucleotides, we call those DNTPs. You have to have a set of short oligonucleotide primers to flank the region of DNA that you want to replicate or you want to be sequenced. And then, of course, you have to have the enzyme that is responsible for catalyzing the dehydration synthesis reaction that builds the nucleic acid polymer. So the issue, though, is that when you're talking about DNA replication that is happening inside of cells, we're going to need several extra enzymes. Number one, the thing to consider is that when you are replicating DNA in a test tube, the way that you kind of unpeel the two strands apart from each other so that you can replicate them separately is that you use high heat. Well, intact living cells can't really tolerate high amounts of heat and high temperatures, so we have to have an enzyme in order to unwind the DNA for us. And we have to have several enzymes to do several other things as well. So DNA replication inside of living cells is going to require a couple of extra enzymes, and these enzymes are DNA helicase, DNA topoisomerase, RNA primase, and DNA ligase. So RNA primase, that is not a typo. It should not be DNA primase. It is RNA primase, and you'll see why here in just a minute. Okay, the picture that you're looking at here shows DNA replication in process. So on this side of this enzyme you see here called topoisomerase, you have the two original strands of DNA that are still together. And then on the left side, you have what is called the replication fork. So this is the portion of the DNA that is actively being replicated. And then to the left of that, you can see the two original strands have separated apart, and you can see we are actively synthesizing new complementary strands for each template strand. So we'll go ahead and cover this step by step in terms of how we actually do this in prokaryotes. We're going to start with a conversation on prokaryotic DNA replication, and we'll cover the differences with eukaryotic replication later on. So first, the DNA is going to be unwound. It should say unwound, not wound. So the DNA is unwound in both directions at the origin of replication of the bacterial chromosome. So remember, a bacterial chromosome is a big circular molecule, and it has one origin of replication. So this unwinding is facilitated by the ATP hydrolyzing enzyme called DNA helicase. So you can see that little triangle right here. DNA helicase's job is to unwind the DNA at the replication fork so that we can then obtain our two single-stranded template strands, and then we can start making complementary strands on top of it. And then you're going to notice here that we have a couple of proteins that bind to the single-stranded DNA called single-stranded binding proteins. They are there to maintain the stability of single-stranded DNA. And you'll recall that DNA doesn't really ever want to be single-stranded, so we have to have these proteins to kind of hold it in place. Now, one issue that we run into is that unwinding the DNA like this causes the double-stranded DNA on the other side of the replication fork, so over here, to become supercoiled. So supercoiling is basically what happens if you have a rubber band on hand. You can actually illustrate this nicely. If you kind of twist the rubber band in your hand, you can kind of see what supercoiling is like. So when DNA becomes supercoiled, it becomes excessively twisted, and this distorts the three-dimensional structure of the nucleic acid, which is actually not a good thing. So this enzyme that you see here called DNA topoisomerase, this helps to relieve the supercoiling by temporarily breaking the backbone of the DNA and then using that in order to relieve the torsion and the tension. And then another issue that we run into here is that since in the laboratory setting we said that we need primers to bind to the DNA, since we don't have those primers just floating around on their own inside of the cell, we need an enzyme to synthesize these primers. The idea here is that DNA polymerase, the enzyme that catalyzes the extension of DNA polymerase cannot synthesize DNA de novo, meaning from nothing. So DNA polymerase will only bind to DNA at what we call a primer template junction. So it will not bind to single-stranded DNA, which is just the template. It will only bind to the template DNA if there is a primer sitting on top of it. So this enzyme RNA primase actually lays down very short RNA-based primers on top of the template strand, and that's going to serve as the binding site for DNA polymerase to then add free nucleotides to the 3' end of the RNA primer so that we can use that as our jumping off point for extending and synthesizing DNA. Okay, before we go any further, let's talk about that complication that we talked about before. So because the two strands are anti-parallel to one another, but because they both have to be replicated from 5' to 3' end, like we said, this is going to present some problems for us. So when the two strands are unwound, we're going to refer to these two strands as the leading strand and the lagging strand. We're going to see here that synthesis of the complementary strand for the leading strand is actually fairly simple, but doing the same thing for the lagging strand is actually a little bit of a pain. So when the DNA polymerase binds to the primer template junction on the leading strand, because the synthesis is going from 5' to 3' towards the outgrowing DNA replication fork, DNA polymerase is able to add free nucleotides and just continue to follow the DNA helicase enzyme as it unwinds the DNA in front of it. So in this case of the leading strand, DNA polymerase is able to extend the DNA in a continuous manner without ever really having to stop. But because the lagging strand, the extended DNA is going to go away from the replication fork, we ensure pretty much that DNA polymerase is always going to run into temporary obstructions that is going to have to cause it to stop and then fall off and then reattach once the replication fork extends a little bit further out. So because of this, primer extension cannot happen continuously. DNA polymerase is always going to have to start, stop, start, stop, start, stop. So we're always having to hit the pause button here to allow the helicase to extend the primer template junction, or to extend the replication fork further out so that we open up a little bit more single-stranded DNA. So because of this, DNA replication happens discontinuously here, and the replication that we are able to do appears as what we call short Okazaki fragments. So instead of laying down a single long continuous piece of DNA, we lay it down in fragments that we call Okazaki fragments. Okay, so another thing to consider is that DNA polymerase enzymes have what is called proofreading exonuclease activity. We talked before in Chapter 10 and Chapter 11, especially when it comes to mitosis, that it's really important that we replicate the DNA faithfully. So basically what DNA polymerase does is it's fairly accurate. It usually does a pretty good job of laying down the correct nucleotide so that it's complementary to what's on the template strand. But every now and then it will make a mistake. And every time DNA polymerase lays down a nucleotide, it is able to check its work. If it made a mistake, it uses this exonuclease activity to remove that nucleotide and then replace it with the correct one. So the major DNA polymerase that does this as it actively synthesizes DNA is DNA polymerase 3. But on top of that, another DNA polymerase that we'll see soon, called DNA polymerase 2, also uses this activity to remove the RNA primers and then replace them with their DNA equivalents. So we don't want there to be RNA in our DNA. And then any gaps that exist between nucleotides get sealed together by the enzyme DNA ligase, which is able to seal the edges and the gaps between any breaks in the phosphodiester backbone of both strands. Okay, so that, in essence, is the DNA replication process. If you can get past the complication that the lagging strand causes for us, it's really not too terrible of a process in terms of keeping track of all the bits and moving pieces. So there are at least 4 or 5 known DNA polymerase enzymes in prokaryotes. DNA polymerase 3 is the primary enzyme involved in DNA replication, and it is usually the one that is going to add nucleotides onto the growing strand. DNA polymerase 1, I screwed up before. I said DNA polymerase 2 does this, but DNA polymerase 1 removes RNA primers and fills in the gaps left behind after the RNA primers are removed. So DNA polymerase 2, what it actually does, to correct my mistake, DNA polymerase 2 is actually a lot more involved in DNA repair, which is something that we will see in the next video. And then a very interesting thing is that DNA polymerase 4 is actually a highly error-prone DNA polymerase. It makes lots and lots and lots of mistakes. It's like someone who's a really bad typist, someone that makes lots and lots of mistakes that always need to be corrected. This might seem counterintuitive. You might be thinking, well, why do we want a DNA polymerase that makes lots of mistakes? Well, you might be surprised to hear the mistakes are actually the whole point. We want this DNA polymerase to make lots of mistakes. It's because this DNA polymerase is only going to be active when the prokaryote is under a lot of stress, like if it isn't able to get enough nutrients or if it's in the presence of an antibiotic. The idea here is that when a bacterium is put under stress, the bacterium will switch over to using this type of DNA polymerase that makes lots of mistakes. And the idea is that we want to make lots of errors so that we increase the likelihood that we drive rapid mutation of important metabolic genes that might allow the bacterium to adapt to stress in a short amount of time. So if we change the nature of the information in the genes, then maybe some of those changes would actually be for the better, and maybe that would mean that we increase the likelihood that these cells would actually survive whatever is stressing them out. Okay, so let's bring up a couple of differences between prokaryotic and eukaryotic DNA replication. Like I said, these two processes are pretty similar, but there are a couple of differences that we do want to take note of. So first, we want to kind of take note of the fact that, like we said, prokaryotic genomes tend to just be one singular circular chromosome that has one origin of replication. Eukaryotic genomes consist of multiple linear chromosomes, like we talked about before, and each chromosome tends to have quite a few origins of replication, so hundreds to thousands of them. So what that means is that you're not replicating the DNA from just one point. You're actually replicating it in many different pieces at different points altogether. We, of course, appreciate that the eukaryotic genome is much, much, much bigger, much more vast. It contains billions of base pairs rather than the millions that you see in prokaryotes. One interesting thing that might surprise you is that DNA replication is actually a lot slower in eukaryotes. Prokaryotes do this at a rate of about 1,000 nucleotides per second. We only do it at about 50 to 100 nucleotides per second. A big reason for this is that eukaryotes utilize many, many, many different types of DNA polymerase, and they basically all work together, and this collaboration slows the process down quite a bit. So, despite there being 14 different types of DNA polymerase, the major players that we're going to see are 3 to 5 of them: alpha, beta, gamma, delta, and epsilon. And then finally, a couple of minor differences. Eukaryotes, to remove the RNA primers that RNA primase lays down, they do not use DNA polymerase 1. They use a different enzyme called RNase H to do this. And then instead of using the sliding clamp protein to hold the DNA polymerase on the growing strand, eukaryotes use something similar called pCNA in order to do this. Okay, so let's talk about one more important thing pertaining to the difference between prokaryotic and eukaryotic replication. Because prokaryotic chromosomes are circular in nature, we don't have to deal with the issue of what to do with the ends of chromosomes, either the 5' end or the 3' end, because if the DNA is a circle, there is no 5' or 3' end. It's just one big continuous circle. So the issue with eukaryotic chromosomes is that since they are linear, they have free 5' and 3' ends. Now you can kind of see where we're going with this, but because of the way lagging strand synthesis works, we cannot possibly properly duplicate the very ends of the chromosomes. Because if we try to do this, with every round of DNA replication, these ends of the chromosomes would get shorter and shorter and shorter. The amount of space we're able to replicate would get shorter with each round of replication, and the chromosomes would basically get gnawed or chewed off at the ends. So as an adaptation to this, eukaryotes have developed regions of DNA at the ends of chromosomes called telomeres. Telomeres actually contain repetitive, nonsensical DNA that doesn't code for anything. So it doesn't contain any genes, it doesn't contain any important information for making proteins. This is DNA code that is meant to be lost with every round of replication. So it's basically kind of wasteful DNA that we can basically say, okay, we know we're going to chew off the ends of the chromosome. Here is some extra DNA we can put at the end so that that's not that big a deal. We just don't want to lose protein coding information from the more interior parts of the chromosome. The telomeres are meant to be lost. The coding regions of DNA are not meant to be lost. So early in development, eukaryotes express an enzyme called telomerase, which is a type of it's not exactly like DNA polymerase, but if you want to think of it like that, that's okay. It's a type of polymerizing enzyme that specifically replicates the DNA at the telomeres. So its mechanism is different than DNA polymerase, so it actually can extend the telomeres. But part of the problem is that this enzyme is only active in stem cells, germ cells, and actively developing cells. Meaning that once we are born and once we grow up and reach adulthood, the vast majority of cells in our body actually do not have telomerase anymore. So every time cells in our body replicate, the telomeres get shorter and shorter and shorter. So like we said, since somatic cells do not possess active telomerase, every time our somatic cells replicate, the telomeres become shorter after every cell cycle. So once the telomeres get sufficiently short, meaning that we're really kind of in danger of chewing off the ends of chromosomes that have DNA, that have protein coding regions, these cells are going to take that as a signal to stop going through the cell cycle, and they go through what is called senescence, which is basically kind of a pathological state of cellular aging. So telomere shortening is thought to be a major mechanism by which people kind of start feeling the process of aging in their 70s and 80s. Basically, the idea is that these people have been alive for so long that their cells have gone through the cell cycle so much that the telomeres have just gotten so short that they can't really do it anymore. So a possible explanation for this, or a possible kind of corroboration of this, is that cancer cells are often found to have very short telomeres, but they have found a way to reactivate their telomerase, so they are actually able to keep dividing, because as long as they're expressing telomerase, they're not in danger of chewing off the most critical edges of the chromosomes that contain the protein coding information. So just kind of an on-your-own question to kind of fit in with what we talked about in Chapter 10 and Chapter 11. Do you think telomerase would qualify as a proto-oncogene or a tumor suppressor gene? Something for you to think about. Okay, that's going to do it for this video. It was a little bit of a longer one, so I thank you for your attention. Join us next time. We'll have one more video in this chapter. We'll talk a little bit about the DNA repair process, and then we'll talk a little bit about the nature of DNA mutations, particularly point mutations, which that will actually serve as a jumping off point for future discussions on transcription and translation and how we actually use the DNA code in order to synthesize proteins. Thanks again, and I will see you next time. So long.